This lesson is going to cover Chapter 9 in your Campbell biology book, Cellular Respiration and Fermentation. So remember, we've been talking about organic molecules, and many of those which contain carbon-hydrogen or carbon-oxygen bonds. Now, in order to release the energy from these bonds, we have to have catabolic reactions or break these bonds. So catabolic reactions are going to release the stored energy by breaking down these complex molecules. Or, in other words, they are exergonic reactions. Exergonic reactions create energy. The idea of breaking down these larger molecules uses enzymes. So in order to do this, we're going to have to use enzymes. And the overall process of breaking down these large molecules, releasing energy and making smaller molecules or monomers is known as digestion. Most of these reactions involve oxidation. Remember, oxidation are where electrons are lost. This is where Leo says Gur comes in handy. Remember, we went over this earlier in the semester where I mentioned that Leo is lose electrons oxidation and Gur is gain electrons reduction. So that will help you remember the difference between oxidation and reduction, things that we'll be talking a lot about in the upcoming lectures. So cellular respiration, which we're going to be talking about largely in this chapter, deals with the complete oxidation of glucose. Remember that oxidation is the loss of electrons. The loss of electrons is always balanced with a reduction, the gain of electrons. So these reactions are often referred to as redox reactions. The importance of the electrons is that they carry energy with them. The amount of energy the electrons carry depends on the orbitals that they occupy. Electrons that are further from the nucleus carry the most energy and are the most unstable. Again, redox reactions include both oxidation and reduction. The transfer of electrons that we have during these chemical reactions release the energies that are stored in organic molecules. This released energy is ultimately used to synthesize ATP. So again, in reduction oxidation reactions or redox reactions, when we lose an electron, it's called oxidation. When we gain an electron, it's referred to as reduction. Take a look at the following equation as an example. We've talked about sodium and chloride coming together to make table salt. So they would come together and make table salt, but this is an ionic bond which quickly dissociates in water. When this happens, sodium loses an electron and becomes positive. Chloride gains an electron and becomes negative. Therefore, sodium, the one who loses the electron and becomes positive, is the one who also is oxidized. Chloride, because it gains the electron and is now negative, becomes reduced. In these reactions, the electron donor is called the reducing agent. And the electron receptor is called the oxidizing agent. Let's take a look at a little more complicated example in this figure, and we can see all this terminology come into play. First of all, we have methane over here. This is the molecule or the compound that is going to be the electron donor. So it's going to lose electrons or become oxidized into carbon dioxide over here. And because it is the one that is the donor, methane is referred to as the reducing agent. Over here we have oxygen. Oxygen is going to gain electrons or become reduced. And because it is the one that is receiving the electrons from methane, it is referred to as the oxidizing agent. So putting all of this together with cellular respiration, I already mentioned before that cellular respiration is the complete oxidation of glucose. And so if we put this together with cellular respiration, we're going to have the fuel, which is going to be glucose in most cases. And this is going to be oxidized. I mentioned cellular respiration again is the complete oxidation of glucose. And oxygen is going to be reduced. Let's take a look at the overall equation for the process of cellular respiration. We have glucose plus oxygen, yielding carbon dioxide as a waste product, water, and energy in the form of ATP. So in the process of cellular respiration, glucose here becomes completely oxidized into carbon dioxide, and oxygen is going to be completely reduced. So the goal of respiration is to produce ATP. Remember, I've already told you that ATP is the energy currency of the cell. The cell needs ATP in order to perform many of its functions, and therefore it needs a lot of ATP. So through cellular respiration, we are going to be able to produce ATP. Let's go ahead and take an overall look at the stages of cellular respiration. In order to harvest energy from glucose, we have to have 3 different stages occur during cellular respiration. The first stage is referred to as glycolysis. Glycolysis begins the breakdown of glucose, where glucose is broken down into 2 molecules of pyruvate. The second step is the citric acid cycle. The citric acid cycle completes the breakdown of glucose. And the third and final step is oxidative phosphorylation. Oxidative phosphorylation accounts for most of ATP synthesis. It's going to be powered by redox reaction, and almost 90% of the ATP generated by cellular respiration comes from oxidative phosphorylation. As you use your book to study these different stages, they've shown glycolysis in blue throughout the chapter. Pyruvate oxidation and the citric acid cycle is shown in orange throughout the chapter, and then oxidative phosphorylation is shown in purple throughout the chapter. This is really nice because you can easily follow throughout the chapter with this nice color coding. Let's take a quick overview look at what's happening here. We see that glycolysis occurs within the cytoplasm of the cell itself, generating some ATP. From glycolysis, we're going to go into pyruvate oxidation and then eventually into the citric acid cycle. Both of these steps occur within the mitochondria of the cell, again generating some more ATP. And finally, those electrons are going to be shuffled into oxidative phosphorylation, the final step. This is where we have electron transport and chemiosmosis, where we are going to generate the most ATP in this final step. So most of the ATP generated is going to be in that final stage referred to as oxidative phosphorylation. I mentioned before that some ATP can be formed in glycolysis and the citric acid cycle, and this is going to be done by something called substrate level phosphorylation. Substrate level phosphorylation is a metabolic reaction that results in the formation of ATP by the direct transfer of a phosphate group to ADP. It uses an enzyme, okay? So you can see in this first picture, this first side, it shows an enzyme, and in order to create ATP, another phosphate group is added to ADP. This creates ATP. We see this happening in both glycolysis and the citric acid cycle. In glycolysis, we are going to have glucose being broken down into 2 molecules of pyruvate. Glycolysis means sugar splitting. Glycolysis occurs whether or not oxygen is present. Glycolysis is the first step in cellular respiration, where we will get 2 molecules of pyruvate, and glycolysis occurs in the cytoplasm of the cell. Glycolysis is broken down into 2 different phases. The first phase is referred to as the energy investment phase. In this phase, beginning the breakdown of glucose, it uses 2 molecules of ATP, hence why it is referred to as the energy investment phase. As we continue through to break down glucose, we enter into the second phase, or the energy payoff phase. This is referred to as the energy payoff phase because we are going to form ATP. Again, remember, ATP in this case is formed through substrate level phosphorylation, where ADP is phosphorylated, or phosphate is added to ADP to create ATP. The net is that we have 2 ATPs formed through glycolysis. So 1 molecule of glucose will yield 2 pyruvates, 2 ATPs, and then we will get 2 NADHs, which will become important later on. Here we can see the complete breakdown of glucose into pyruvate, beginning with the energy investment phase. Again, this is the phase which requires ATP as we begin the breaking down of glucose into the separate steps. And then we have the energy payoff phase, the final steps of glycolysis in order to generate pyruvate. And this is the payoff phase. So this is where through substrate level phosphorylation, we are getting ATP from adding phosphate to ADP with the use of an enzyme. So now through glycolysis, we have 2 molecules of pyruvate for every glucose that goes through glycolysis. And the fate of that pyruvate is going to depend on oxygen availability. If oxygen is available, then pyruvate will enter into the mitochondrion. The pyruvate will enter into the mitochondrion for eukaryotic cells, but if we're talking about prokaryotic cells, this is going to happen in the plasma membrane. At this point, glucose will enter in to be completely oxidized. Before this can happen, pyruvate has to be converted to acetyl coenzyme A. Since one glucose is going to yield 2 pyruvates, this process would have to happen twice, so we would end up with 2 acetyl coenzyme A's. So in the presence of oxygen, glycolysis would yield from 1 glucose 2 pyruvates. The pyruvate would enter into the mitochondrion and go through pyruvate oxidation, and this would yield 2 acetyl coenzyme A's. Each pyruvate would have to go through once, so we would end up with 2, and these are going to go into the citric acid cycle, which we will be talking about next. The citric acid cycle is also referred to as the Krebs cycle. This is the cycle which is going to complete the breakdown of pyruvate to carbon dioxide. Remember, pyruvate came from glucose, and so we're going to really be completing the oxidation of glucose during the Krebs cycle. This is going to occur in the matrix of the mitochondria, in eukaryotic cells, but in prokaryotic cells, it's going to occur in the cytoplasm. The citric acid cycle has 8 steps. Each step is going to be catalyzed by a specific enzyme. The first step is going to involve the acetyl-CoA that just came from pyruvate. together with oxaloacetate to yield citric acid, which is citrate. Hence the name of the cycle. This is the first step. The next 7 steps are going to decompose this citric acid back to oxaloacetate, and that's going to make the process a complete cycle. Here we see pyruvate going into acetyl-CoA. Pyruvate has to first be converted into acetyl-CoA before it can enter into the citric acid cycle. Remember again, the first step of the citric acid cycle turns this acetyl-CoA into citric acid, and it cycles all the way back, allowing it to be called a cycle. One turn of the citric acid cycle is going to produce 2 molecules of carbon dioxide. Okay, so we can see this going through one turn, 2 carbon dioxide, 2 molecules of carbon dioxide. We are going to have 3 NADs that are going to be reduced to NADH. We're going to have 1 FAD reduced to FADH, 2. Okay, and then it's going to produce 1 ATP. Because 1 glucose is going to yield 2 pyruvates, which are going to yield 2 acetyl-CoAs. For each glucose, we're going to have 2 turns of the citric acid cycle. And so we would have to multiply all of these by 2 in order to get how many we would receive from 1 glucose molecule. So let's go ahead and look at an overview of the citric acid cycle. Again, the citric acid cycle begins with pyruvate turning into acetyl-CoA. Acetyl-CoA then enters into the citric acid cycle. Acetyl-CoA with oxaloacetate is going to yield citrate or citric acid. That is the first step, and the reason why the citric acid cycle is referred to as a citric acid cycle. As we continue through the steps, we will see the breakdown of citric acid. Through this process, we will generate some carbon dioxide and some NADH, ATP through substrate level phosphorylation, FADH2, and NADH. The cycles back to the beginning again, where we get oxaloacetate, which again is why it's referred to as a cycle. So now we've finished with the citric acid cycle, and we've gone through glycolysis and the citric acid cycle, and we've generated some ATP through substrate level phosphorylation. But most of the energy that has been extracted from food is now in the form of NADH and FADH2, which I've been referring to. NADH and FADH2 are electron carriers. These two are going to donate their electrons to the electron transport chain. The electron transport chain is a series of membrane-bound electron carriers that are embedded in the inner membrane of the mitochondrion. Remember that this inner membrane of the mitochondrion is referred to as cristae. We see this process happening at the cristae of the mitochondrion in eukaryotic cells. If we are talking about prokaryotic cells, this process is occurring in the plasma membrane. So the electron transport chain is going to occur in this last step where we have oxidative phosphorylation. During the electron transport chain, which is again a series of membrane-bound electron carriers that are embedded in the mitochondria, we are going to have electrons that are passed from carrier to carrier throughout the membrane, and as they pass the electrons, they are going to drop in free energy as they go down the chain. Eventually, it's going to go to the last electron acceptor, which is oxygen in this case, and then finally, water is going to be formed as a byproduct of these reactions. As you can see through this process, ATP is not generated directly by the electron transport chain. ATP is going to be formed by a process referred to as chemiosmosis. As we go through the electron transport chain and the electrons are transferred from one membrane-bound carrier to the next, what's going to happen is this is going to cause proteins to pump hydrogen ions from the mitochondrial matrix to the intermembrane space. This process establishes a proton gradient. The mitochondrial matrix is negative compared with the intermembrane space. So this allows the positively charged protons to be attracted to the matrix. The higher outer concentration of protons also tends to drive the protons back by diffusion. Hydrogen ions are then going to move back across the membrane, passing through a protein complex known as ATP synthase. This is an enzyme that uses energy of the gradient to catalyze the synthesis of ATP from ADP and phosphate. This whole process is referred to as chemiosmosis, basically using the energy of the hydrogen ion gradient to drive cellular work. The hydrogen ion gradient is referred to as a proton motive force. This emphasizes its capacity to do work. Let's take a look at how this happens. As electron transfer is happening in the electron transport chain, this causes proteins to pump hydrogen ions into the intermembrane space, which is up here. So we have a lot of hydrogen ions that are coming out into the intermembrane space as the hydrogen ions then pass back into the mitochondrial matrix. One, because the mitochondrial matrix is negative, and two, because there ends up being a higher concentration of hydrogen ions in the intermembrane space, allowing them to move from a higher concentration to a lower concentration. They will pass back in through ATP synthase. As they pass back in through this ATP synthase, it is going to generate ATP by phosphorylating ADP. Here is a larger picture of what's happening. In this first part, we can see the electron transport chain as we pass electrons from membrane receptor to membrane receptor. Hydrogen ions are pumped through into the intermembrane space. Eventually, the concentration of hydrogen ions gets greater in the intermembrane space than in the mitochondrial matrix, which is below. And so hydrogen ions are then going to pass into ATP synthase to get back into the intermembrane space. And as it does so, it's going to phosphorylate ADPs and make them into ATPs. This is the way that the majority of ATP is created throughout cellular respiration. The process of the electron transport chain and chemiosmosis, which is the final part here, the entire process of both of these together is referred to as oxidative phosphorylation. So a recap on cellular respiration. Initially, we have to have glycolysis, seen in the blue box. That's the first step that has to happen, where glucose is broken down into 2 molecules of pyruvate. This yields about 2 ATPs. Pyruvate then enters into the mitochondria, where it is broken down into acetyl coenzyme A. Since there are 2 pyruvates, we will get 2 acetyl coenzyme A. Acetyl coenzyme A is going to enter into the citric acid cycle, where we will get about 2 molecules of ATP in this process as well. And then finally, all the electrons are transferred from NADH and FADH2 into the electron transport chain and chemiosmosis, which is the final step of oxidative phosphorylation. In this process, we will get about 26 to 28 molecules of ATP, being the area where the largest amount of ATP is going to be generated. So altogether, we get about 30 or 32 molecules of ATP for 1 molecule of glucose during cellular respiration. So the process we just talked about uses oxygen. So in order to go through the Krebs cycle and the electron transport chain, we're going to need oxygen in order for that to happen. But now, what happens if there is no oxygen? If there is no oxygen, then glycolysis. Remember, glycolysis can happen with or without oxygen. So if there is no oxygen, then glycolysis is going to couple with either anaerobic respiration or fermentation. Anaerobic respiration is going to use an electron transport chain with the final electron acceptor being something other than oxygen. These molecules are inorganic molecules. And an example of that would be sulfate. Anaerobic respiration is done by most bacteria and some yeast and fungi. And with this process, fewer ATP are produced than with aerobic respiration. Anaerobic respiration can be done by what are called methanogens. In this process, methanogens use carbon dioxide. And carbon dioxide is then reduced to methane. Anaerobic respiration can also be done by sulfur bacteria. And in this case, they use inorganic sulfate. And they're going to reduce that to hydrogen sulfide. So it just depends what type of bacteria you're talking about, but then their products are going to be either methane or hydrogen sulfide. Fermentation uses organic molecules as the final electron acceptor, and an example of this would be pyruvate or acetaldehyde. There are two different types of fermentation that we can talk about. The first type is alcohol fermentation. In alcohol fermentation, pyruvate is converted to ethanol in 2 steps. The first step involves releasing carbon dioxide. And in the second step, we have ethanol produced. Alcohol fermentation is used by yeast in brewing, winemaking, and baking. The second type of fermentation that we can talk about is lactic acid fermentation. In lactic acid fermentation, pyruvate is reduced by NADH. And this forms lactic acid or lactate as the end product. And in this fermentation, there is no release of carbon dioxide. Lactic acid fermentation is used by some fungi and bacteria to make cheese and yogurt. You've probably also heard of lactic acid buildup in muscles. Human cells can use lactic acid fermentation to generate ATP when there is a lack of oxygen. So, for instance, if you start running and your muscles use up all the oxygen that's in the muscles in order to create ATP, then it will convert to lactic acid fermentation where you'll start to feel a cramp because of the lactic acid buildup. If you continue to run and you increase your respiration rate and more oxygen comes into the body and goes to those muscles, then lactic acid fermentation will stop and cellular respiration, aerobic respiration will then continue. So the muscle cells have this ability to do lactic acid fermentation in order to keep up with the production of ATP so that the muscles can continue to contract. This figure shows the two types of fermentation side by side. On the left-hand side, you'll notice alcohol fermentation, where we use pyruvate to eventually make ethanol. On the right-hand side, in lactic acid fermentation, pyruvate is used to make lactate or lactic acid. Again, this depends on the organism and its ability to either produce alcohol or lactic acid. Let's do a quick comparison of fermentation with anaerobic respiration. As we spoke about before, cellular respiration or aerobic respiration produces 32 ATPs. That is by far the best way to produce ATP. We're going to get the most from that. Fermentation and anaerobic respiration make a net of 2 ATPs. If we talk about anaerobes, we can separate organisms based on whether they utilize oxygen or don't utilize oxygen. So aerobic organisms or aerobes will use oxygen. Anaerobes will mostly use no oxygen. However, we can separate anaerobes into two different categories. Obligate anaerobes are those organisms that cannot survive in the presence of oxygen. So these will carry out fermentation or anaerobic respiration solely. The other type of anaerobes, facultative anaerobes, can survive with oxygen or without oxygen. So they are actually capable of doing either fermentation or cellular respiration. So these will be different types of bacteria that are actually capable of switching and living in the presence of oxygen or without oxygen. The figure you see here kind of sums up this idea, starting with glucose and going through glycolysis. Remember, glycolysis can happen with or without oxygen, so we always get glycolysis forming a pyruvate. Remember, the fate of pyruvate is going to depend on whether or not oxygen is present. If oxygen is present, it's going to be able to go through aerobic or cellular respiration, eventually forming acetyl coenzyme A, going into the Krebs cycle, and so forth. If oxygen is not present, then fermentation or anaerobic respiration is going to happen. And we're going to have the production of things like ethanol, lactate, or other products depending on that organism and its capabilities. All right, let's talk about the versatility of catabolism. Catabolic pathways funnel electrons from many kinds of organic molecules into cellular respiration. That means we do not only need glucose in order for cellular respiration to occur. However, glucose is the best and it's going to make the most ATP. Glycolysis can accept a wide range of carbohydrates, again, not just glucose. Proteins can become part of cellular respiration, but they have to be broken down into amino acids. Remember, they're monomers. And then after they're broken into their amino acids, different amino groups can actually feed glycolysis or the citric acid cycle. Fats can be broken down into glycerol. Remember again the monomers. Glycerol can actually be used in glycolysis. And the other part of fats are fatty acids. So if we again break them into their monomers, fatty acids can be used in generating acetyl coenzyme A. The figure here shows you these examples. At the very top, we can see different types of organic molecules, proteins, carbohydrates, and fats. Proteins broken down into their monomers, amino acids, carbohydrates into their monomers, monosaccharides, which are sugars. Remember, it doesn't just have to be glucose, though glucose is going to yield the most. Fats would then be broken down into glycerol and fatty acids. The body can then utilize these things into different parts of the cycle, some of them entering into glycolysis itself, while others can enter some types of amino acid groups. Amino groups can go into the citric acid cycle. So we can see these can be put into many different parts of the cycle. So if the body runs out of glucose, it can break down things like proteins and fats or different carbohydrates and put them into different parts of these cycles to help continue to generate ATP even if there is no glucose there. Of course, glucose is going to generate the most. So we've been talking the majority of this lecture on catabolism, which is breaking things apart. What if we talk about putting things together, or biosynthesis, okay? In biosynthesis, the body uses small molecules to build other substances. These small molecules may come directly from food, from glycolysis, or from the citric acid cycle. In order to build things up, this uses energy, and so it is referred to as an endergonic reaction. Let's talk about the regulation of cellular respiration. Now, in living organisms, all the metabolic reactions that occur in living organisms need to be regulated so that they're not occurring when the body does not need them to occur. Feedback inhibition is the most common mechanism for metabolic control. An example of feedback inhibition for our specific cellular respiration is that if ATP concentration begins to drop, then respiration is going to speed up. However, when there is plenty of ATP available, then respiration is going to slow down. You can see in this figure how feedback inhibition works. In feedback inhibition, oftentimes one of the products of the metabolic reactions can also act as the inhibitor. So in this case, we can see that ATP has the ability to do that. When ATP concentration increases, ATP can actually go back to a step in glycolysis and inhibit it. What this means is it keeps it from happening, and if that process cannot happen, then nothing else after it can happen, which means it's going to stop the citric acid cycle and oxidative phosphorylation as well. However, when ATP levels drop, if they drop, then this removes the inhibition and allows glycolysis to proceed, which would then allow the citric acid cycle to happen, oxidative phosphorylation to happen, and more ATP to be created. As you see how this process works, this will keep ATP levels constant in living organisms rather than them having too much or too little. Other things from the citric acid cycle, like citrate, can also go back and inhibit as well. When levels of citrate get high, it will go back and inhibit steps in glycolysis. When citrate levels get low, then it will not be there to inhibit anymore, and the process can continue.